Back to another session of biochemistry classes. So in the last class, if you can remember, we finished with the DNA replication, right? So this class will go on to the other molecular biology method, that is transcription, right? So before we go on to what is transcription, we should understand what is, we call it as central dogma of molecular biology. So in the last class, what did we study? We studied about replication, that is a single DNA strand is taken and it is copied again and it is synthesized, right? So that process is almost similar to how we take a Xerox copy of one page from a book. So whatever we want, we take that as a copy, right? So that is called as replication. Now this DNA acts as a template and it leads to the synthesis of mRNA. Okay, so this process we call it as transcription, right? So when this RNA is synthesized, it uh, enters into the cytoplasm and it codes for the amino acids and it leads to the synthesis of proteins. So this process we call it as translation, right? So this is the central dogma of molecular biology. So today's class will go on to understand the process of transcription. Okay, so what is transcription? It is a process in which the RNA is synthesized from DNA and the product that is formed, it is the known as a primary transcript. So this primary transcript has to be undergo a lot of changes to form the mature or the active RNA, and this we call it as a post-transcriptional modification. Okay, so whatever is going to be formed first from transcription, we call it as a primary transcript, and this primary transcript undergoes certain post-transcriptional modification to form the mature or the active RNA. Okay, so transcription, we will study this topic under two headings, that is, one is transcription in prokaryotics and transcription in eukaryotics. So in today's class we will talk only about transcription in prokaryotes. So when we talk about transcription in prokaryotes, you should know that there are important enzyme that participates in transcription. So we call this enzyme as DNA dependent RNA polymerase enzyme, okay, which is required for the synthesis of all the types of RNA. So we know that from the chemistry part that there are ribosomal RNA, then you have the tRNA, and then you have the HNR RNA. Okay, so what is it? All the RNA synthesis will require DNA dependent RNA polymerase enzyme. So this DNA dependent RNA polymerase enzyme, we call it as a hollow enzyme, which is made up of 5 polypeptide subunits. Okay, so that is 2 units of alpha subunits, 1 unit of beta subunit, 1 unit of beta dash subunit, 1 omega, and 1 sigma factor. Okay, so it is made up of 5 polypeptide subunits, right? So what is the importance of the sigma subunit? It is helps in recognizing and fixes to the on the promoter site. And what does the alpha does? Alpha, it detects the DNA to be transcribed. Okay, and then you have the beta one, which helps in polymerization, and the beta dash, it helps in unwinding of the DNA strand. Okay, so what is it? It is, you require a DNA dependent RNA polymerase enzyme, and it is required for synthesis all the different types of RNA. It is an hollow enzyme with 5 polypeptide subunits, 2 alpha, 1 beta, 1 beta dash, 1 omega, and 1 sigma. The alpha unit is helpful in detects the DNA that has to be transcribed. The sigma subunit recognizes and fixes on the promoter site. The beta polymerizes. The beta dash helps in unwinding of a DNA. Okay, so we'll look into the how the RNA polymerase looks. It's an hollow enzyme. We said it is made up of 5 subunits. Okay, this is the sigma subunit, 2 alpha, 1 beta, 1 beta dash, and 1 omega subunit it requires. So this is a hollow enzyme, okay, RNA polymerase, which is dependent on the DNA. Okay, so transcription can be broadly studied under 3 headings, that is initiation, elongation, and termination. Okay, so in initiation we have a number of other steps. We will talk about the initiation first. Okay, so what is it? As we said in the how the sigma factor of the RNA polymerase, it helps in identification of the promoter sites. Okay, so when we talk about the promoter sites in the prokaryotics, you have 2 promoter sites. Okay, so we call this 2 promoter site, one as a Tata box, or we also called it as primrose box. Okay, so this is having -10 base pairs upstream from the starting point of the transcription. Okay, that is before the transcription site where this starts. Okay, so that is the primrose box, or we call it as the Tata box. And then you have the 35 sequence base pairs. Okay, that is made up of TGTT-GACA base pairs, and which is located about -35 base pairs from the starting point. So now what is it? In the initiation, first the sigma factor of the RNA polymerase enzyme recognizes the promoter site for the initiation of transcription. So in the prokaryotics, you have 2 types of promoter site, that is the primrose box, that's the Tata box, which has 10 base pair, 10 -10 bases upstream from the starting point of the transcription. And then you have the 35 sequence base pair, which is located about -35 bases from the starting point of transcription, right? So when you talk about initiation, you also have to know under that the promoter, as we spoke now about the primrose box or the Tata box and the 35 base pairs. And then you have the transcription unit, the upstream and the downstream sequences, the template and the coding strands. Okay, so under the steps you have the closed, then you have the open, and then you have the transcription bubble that is formed and the promoter clearance. These are the steps. Okay, so the closed DNA strands are opened up, the transcription bulb is formed, and then the promoter clearance takes place in initiation. Okay, so first we'll go on to, so in initiation, the DNA molecule, that is the template strand, that is from the 3' to the 5', it is the template strand, and the 5' to the 3' is the coding strand. And there are certain regions on the coding strand upstream to this region, we call it as the promoter site, okay, or the promoter region. So that is about 5', 3' direction, and the direction is always in the transcription unit. Now the RNA polymerase recognizes the promoter site or the promoter region. There are -10 base pair, that is the Tata box or the primrose box, and -35 base pairs, that is the initial site for the RNA polymerase to bind. Okay, so the DNA molecule, that is the template, is always 3', 5'. The coding strand is from the 5' to the 3' strand, and the coding strand contains certain promoter region or the promoter site. Okay, and the RNA polymerase recognizes the promoter sites, that is -10 Tata box, or it is the -35 base pairs, and it binds to the promoter site which is initiated by the RNA polymerase. Now this RNA polymerase first attaches to the -35 base pair sequence, and it initiates the separation of the DNA strand. So when it separates the DNA strand, the local unwinding in the region happens of the Tata box, and this Tata box, when it unwinds, it leads to the formation of an open complex. Okay, so the initiation of synthesis leads to synthesis about 5 to 10 base pair by the RNA polymerase, and this opening is about 20 to 20 base pairs, and we call this as a transcription bulb. Okay, so when this transcription bulb forms, now what happens? The RNA polymerase will move forwards towards the transcription site. So what is it? It leaves the promoter site and it moves forward towards the transcription site, and this process we call it as a promoter clearance, right? So what is it? First, the RNA polymerase attaches to the -35 base pair, and this leads to separation of the DNA strand that causes local unwinding in the region of the Tata box. The closed box leads to the formation of open complex. This initiates to the synthesis of about 5 to 10 base pairs by the RNA polymerase, and the opening leads to about 20 base pairs, and this area we call it as a transcription bulb. So when this transcription bulb is formed, the RNA polymerase, after it synthesizes there, it moves forward towards the transcription site, and this process we call it as a promoter clearance, right? So in the next slide, we'll just have a look at what happens here. So this is the RNA polymerase enzyme, which is the hollow enzyme made up of 5 polypeptide chain. It binds to the promoter site. It recognizes the promoter site and it binds to the promoter site, as you can see here. So it binds to the DNA strand. So this is now the closed complex. So it binds to -35 base pairs and -10 base pairs, okay, and it forms the closed complex. Now what happens here? The RNA polymerase now leads to the unwinding of the DNA strand, that is the closed complex gets converted into the open complex, right? And thereby the base pairs are formed, and what happens? The transcription bulb is formed, which leads to the formation of about 20 base pairs here, and there is locally what happens? The RNA polymerase from the promoter site, it moves towards the transcription site, and we call this process as the promoter clearance, right? So in the next slide you can have a look. So initiation with promoter clearance. So what happens here? The RNA polymerase enzyme is attached to the transcription bulb where it forms the open complex. So what happens here? The sigma factor is also released, and what happens? There is synthesis of the mRNA, as you can see here, and the promoter clearance takes place as it moves from the transcription bulb. Okay, so this is the promoter site, you can see here, right? So this we call it as clearance of the promoter, right? So RNA polymerase moves towards the transcription site. So then this is the whole process of initiation. Next we'll go on to elongation. So in elongation, the first step is polymerization, that is the nucleotides are added to the 3' hydroxyl group, about 5 to 10 base pairs. So this leads to the formation of phosphodiester linkages. And then RNA polymerase also does proofreading activity, and then you have a formation of a protein bridge in the 3' 5' template strand of the DNA. So here the deoxynucleotides are present. The newly synthesized RNA molecule is also present. Now what happens when this is formed? There is a protein bridge which is attached to the DNA template. So this protein bridge gauges the synthesis of the new synthesis of the new RNA strand, okay, by keeping the DNA as a template. So it sees to that only one nucleotide is available for the synthesis of another new RNA strand. So only single each nucleotide is added after one each other. So this protein bridge will prevent multiple addition of more than one nucleotide base pair in the new synthesized RNA strand. Okay, so this is the importance of the protein bridge. So in the next slide you can see how this phosphodiester linkage is formed by the nucleotide. The 3' end hydroxyl group attaches to the 5' end phosphate group here in the nucleotide. The RNA polymerase initiates and pyrophosphate is released, and here you can see the phosphodiester bonds formed between the two nucleotide bases. Okay, so this is the formation of phosphodiester bond. So this is the elongation process. Next we'll go on to the termination process. So in termination you have the intrinsic termination and then you have the raw protein dependent termination. Okay, so you have two types of termination, intrinsic termination and raw protein dependent termination. So what do you mean by intrinsic termination? So in this intrinsic termination, you have certain sequences on the newly synthesized RNA molecule from the 5' end to the 3' end, which is complementary to each other. So you can see G, C, G, C, same thing, C, G, C, G, which is complementary. So by this, the some sequences on the template strand also like poly AA, which brings about synthesis of poly GGG, and this will bring about termination of the transcription. So there's this intrinsic termination is nothing but there is synthesis of complementary strand which is in the RNA, newly synthesized RNA and in the DNA. So this will bring about termination of transcription. Okay, so what is the next type of transcription? We said it is the row dependent termination. Okay, so the row protein dependent, it has helicase activity and it also requires ATP. So the template strand, the newly synthesized RNA strand, the row protein binds together here and it cuts the base pairs between the RNA and the DNA, and thereby it brings termination of the transcription. So what is it? This protein dependent, what is it? It has an helicase activity and it requires ATP molecule. So now what does this row protein does? It binds to the DNA molecule and the newly synthesized RNA strand. Okay, and when it binds, it cuts the bases between the DNA and the RNA, and thus it brings about the termination of the transcription. Okay, so here in the next slide you can see here the first one is the intrinsic termination. So you see that the base pairs are complementary to each other and this leads to termination of the transcription. Okay, the complementary base pairs leads to the termination of the transcription and thereby it forms a loop and it initiates and terminates the transcription. Okay, so this is the row independent termination. Okay, so this brings the end of the transcription process. So this transcription of the prokaryotes comes as a long note, and then what is it? First you have to write what is the central dogma of molecular biology. Then talk about the what is RNA DNA dependent RNA polymerase enzyme. You write about all the different polypeptide chain and what is the function, and then write about the all the three processes: initiation, elongation and termination in detail, right? And then you can give a little bit about the termination, and also see to that you draw diagrams about all the three steps in detail, right? So this finishes with um, transcription process in prokaryotes. In the next class, we'll go on to transcription of eukaryotics. Okay, thank you and have a nice day.